Good afternoon to all. In the last class we discussed about the second group of organisms under kingdom Monera, that is called as cyanobacteria. So first of all we discussed what are archaebacteria, and archaebacteria are the oldest group of bacteria originated in the living world. They are of three types: methanogens, halophiles and thermoacidophiles. Methane producing archaebacteria are called methanogens, and organisms, bacteria are living in the salt solutions are called as halophiles, and bacteria living in the hot acid medium are called as thermoacidophiles. There are three groups of archaebacteria. Then comes to second group called cyanobacteria. They are the second group of living organisms originated in the living world. They are the first photosynthetic organisms. They began to produce oxygen by photosynthesis. And another importance is that they have the capacity to absorb nitrogen and convert it into ammonia. That process is called nitrogen fixation, because they have a specialized cell in cyanobacteria like spirulina. There is a specialized cell called a heterocyst helping for nitrogen fixation. And they contain large amount of protein, so they are made into capsules and eaten by the persons who are traveling in the space. So they are the cyanobacteria are important. They produce more oxygen and they become photosynthesis. So actually photosynthesis was not started by plants, but started before that. Photosynthesis was started by the cyanobacteria, and they began to produce oxygen, and that oxygen combined with hydrogen result in the formation of much more amount of water. As a result of that, they began to produce, more amount of water is produced on the earth's surface, which lead to reducing the temperature of the earth's surface, by which new type of organisms originated like protozoa or alga originated. When water is accumulated in the earth's surface, rain started, so the temperature reduced, so it leads to the evolution more faster than the previous. So cyanobacteria are very important. They speed up the process of evolution by reducing the temperature of the earth's surface. So that is cyanobacteria are more important. Then comes the third category called as true bacteria or eubacteria. True bacteria or eubacteria. True bacteria or eubacteria. That is the third group of organisms coming under kingdom Monera, are called as eubacteria or true bacteria. In the textbook some explanations may be different. Don't bother about that, because we cannot mix the two cyanobacteria or archaebacteria, true bacteria together. So the explanation in the textbook is somewhat different, but you can follow this explanation as you like. Then see the true bacteria or eubacteria. This is the third group of bacteria coming under the kingdom Monera. And this true bacteria originated after the last group of organisms in the kingdom Monera, after cyanobacteria or along with cyanobacteria. They are the organisms originated then after. And these true bacteria are now present in the universe more in number. They affect the human beings positively and negatively. Various types of bacteria are there, true bacteria. And these true bacteria are having typical structure, prokaryotic cell structure, without nucleus, without membrane bound organelles are there. So the structure is typical prokaryotic, and maybe most of them are unicellular. And based on the nutrition, we can classify the true bacteria into two categories. Based on the nutrition. What is nutrition? How they are getting food material. Based on that, true bacteria were classified into two categories. Some are called autotrophs. Some are called heterotrophs. Do you know what is the meaning of autotrophs and heterotrophs? Autotrophs are organisms which are preparing food material by themselves are called as autotrophs. See, organisms which are preparing, which are producing food material by themselves are called as autotrophs. And the opposite meaning is heterotrophs. Heterotrophs means organisms which cannot prepare food material by photosynthesis, which cannot prepare food, or which depend upon some other organisms for getting food material. Because heterotrophs cannot prepare food like autotrophs. They cannot do photosynthesis. they cannot produce food material. So how they are getting food material? They are getting food from other organisms. So they are called as heterotrophs, organisms which cannot prepare food material, which are feeding on, which are getting food from autotrophs are called heterotrophs. So the bacteria are of two types. Some of the bacteria are preparing food by themselves. They are called as autotrophs. Some cannot prepare food material. They are called as heterotrophs. So bacteria based on the nutrition classified into autotrophs and heterotrophs. We can see autotrophs. Autotrophs are again classified into two categories. That is called photoautotrophs and chemoautotrophs. See, photoautotrophs and chemoautotrophs. Photoautotrophs means, you know what is the meaning of photo? Sunlight. Sunlight. Photo means sunlight. So what is photoautotrophs? Organisms which are preparing food material by absorbing solar energy or sunlight are called photoautotrophs. They are similar to plants. You need to see plants. Plants have chlorophyll. Bacteria have bacteriochlorophyll. So chlorophyll present in the bacteria are called bacteriochlorophyll, bacterioviridin. Like that, bacteria contain certain pigments. Coloring substances are called as pigments. So, these photoautotrophic bacteria have the pigment color. They absorb solar energy or sunlight. They prepare food by photosynthesis. They prepare food by using sunlight or solar energy. So, such bacteria preparing food or absorbing sunlight are called photoautotrophs. Second category is called chemoautotrophs. Chemo means chemical energy. Some of the bacteria cannot absorb sunlight. They cannot use solar energy. So how they are getting energy? They are getting energy from chemical substances. Such bacteria using chemical energy are called as chemoautotrophic bacteria. So both are preparing food material, but what is the difference? Source of energy is different. This bacteria get the solar energy from the sunlight is called solar energy. This bacteria use energy from chemical substances called as chemical energy. Anyhow, preparation of food materials require energy. There are different sources of energy. So like that, bacteria absorbing energy, solar energy are called photoautotrophs. Similar to plants, bacteria using energy from chemical substances are called chemoautotrophs. And in the chemoautotrophs, various examples you have seen. There are certain bacteria called as, you can study later on, nitrifying bacteria. There are called sulfur bacteria. There are called ammodifying bacteria. These are all bacteria. Example for nitrifying bacteria. You can study later on. Sulfur bacteria obtaining energy from compounds of sulfur. Ammodifying bacteria obtaining energy from nitrogen compounds, ammonia-like compounds. So like that, these are all getting energy from chemical substances. Such bacteria are called chemoautotrophs. So here two categories. Then comes heterotrophic bacteria. Heterotrophic bacteria means what? Bacteria which cannot prepare food. We are getting energy from food from some other living organisms are called as heterotrophs. Heterotrophs are again categorized into two groups. Some are called parasite. 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 Some are called saprophyte. Saprobe or saprophyte. What is parasite? Bacteria which are absorbing food from living organisms are called as parasite. Organisms absorbing food from living organisms are called parasite. And organisms absorbing food from dead organisms' body are called as saprophyte or saprobe or decomposers. See, the difference is that both the parasite and saprophyte cannot prepare food material. They have no pigment. They cannot absorb energy. How they prepare food material? Some of the bacteria are getting food from living organisms' body. Living from our body, living inside plant body. living inside animal body and taking food material. Such bacteria absorbing food from living organisms body are called a parasite. Some other bacteria absorb food from dead body of organisms, dead body of plants and animals. Such organisms are called saprophyte, or they are called decomposers. They are decomposing the dead body of plants and animals. After the death of plants and animals, the body is buried in the soil. They are decomposing. Bacteria and fungus are called decomposers. So, remember that heterotrophs are of two types. Absorbing food from living organisms body are called parasites. Absorbing food from dead organisms body are called saprophyte. And parasites are there. Various examples are there. For example, you might have heard about a bacteria called Vibrio cholerae. See? Vibrio cholerae. And what is the importance of bacteria? Vibrio cholerae, causing cholera in human beings, living inside our body, absorbing food from our body, causing diseases. Salmonella typhosa, bacteria causing typhoid. These are all parasites, living in our body, absorbing food from our body, causing diseases in our body. Such organisms are called parasite. Some are called saprophyte. They are decomposers. They are absorbing food from dead organisms body. For example, pseudomonas bacteria, highly decomposer, absorbing food from dead organisms body. Like that, bacteria based on the nutrition classified into two types. Differentiate between, the question is that write the difference between autotrophs and heterotrophs. See, another question, write the difference between photoautotrophs and chemoautotrophs. Another question, write the difference between parasite and saprophyte. Give example. So this is an important one. You can take out this while you are writing the notes, and I will give the teaching note. I will give the notes later on. Okay? Bacteria were classified into autotrophs and heterotrophs. Then another point, we have to explain what are the positive and negative effect of bacteria on human beings. Bacteria are great in number. They affect human beings greatly, positively and negatively. See, what are the effects of, discuss the effect of positive and negative effect of bacteria. Negative and positive effect of bacteria. What are the uses of bacteria? Uses are called positive effect. What are the positive effect and negative effect of bacteria? Positive effect. What are the positive effect? What are the uses of bacteria? Bacteria are very useful to human beings for various purposes. We can point out one by one. First of all, bacteria called as lactobacillus. You know the bacteria called lactobacillus bacteria. This lactobacillus bacteria convert milk into curd. Very important in the production of milk industry. Production of curd. You know that we take little milk and putting curd in that, it will be converted completely. That process is called lactobacillus bacteria will do the process. Lactobacillus bacteria. Lactic acid, no, lactose is the sugar present in the milk. So this bacillus bacteria, lactobacillus bacteria is used in the milk industry for the production conversion of milk into curd. Second importance is that they are main decomposers of the soil. They are main decomposers. For example, the bacteria like pseudomonas. They are strong decomposers of the soil. You know that the dead body is of the plants and animals are completely buried in the soil after the death. What happens to the dead body? After some days, the body is completely broken into pieces. That process is called decomposition, and that helps in the recycling of materials that we can discuss later on. Decomposition the second year. If the dead body is buried in the soil, if they are not acted upon by bacteria, it cannot be broken into pieces. So fungus and bacteria mainly help in the decomposition. The process of breaking the body components is called as decomposition. And if there is a recycling of materials that you studied in your school syllabus, water cycle, carbon cycle, etc. That is why these bacteria will break. Our body is mainly made up of carbon, hydrogen and oxygen. From where we are getting this one? Carbon enters into our body from atmosphere, oxygen enters into body, hydrogen enters into body. That after the death and decomposition, this carbon again reaches to atmosphere, hydrogen again reaches to atmosphere. So the body was broken by decomposers. Those decomposers are helping they decompose the dead body of plants and animals and help in the recycling of materials. Then third one, some of the bacteria like rhizobium, rhizobium. Rhizobium bacteria help in the process of nitrogen fixation. You know, just already said that cyanobacteria are doing nitrogen fixation. What do you mean by nitrogen fixation? The process of conversion of nitrogen into atmospheric nitrogen into ammonia is called nitrogen fixation. There is an example called rhizobium bacteria, where it is living in the root nodule of leguminous plants, beans or pea-like plants. So in the root nodule, I said that root nodules are the root of the pea plant. If you are taking the pea plant or beans plant, the root contains swellings like that. These swellings are called what? Root nodules. Inside the root nodule, there is a bacteria called Rhizobium radicicola. Rhizobium. That rhizobium helps to fix the nitrogen. It absorbs nitrogen and converts it into ammonia. The process of conversion of nitrogen to ammonia is called nitrogen fixation. So the bacteria will do nitrogen fixation. So bacteria are doing nitrogen fixation. So that is very important in the agriculture. What is happening? Nitrogen is converted to ammonia. So that what happens? These bacteria are living in the soil. So what happens? The ammonia content of the soil increases. The nitrogen content of the soil increases. So that is important for agriculture, improving the fertility of the soil. So bacteria are very important too. Rhizobium like bacteria are helping in the improving the fertility of the soil and helping in agriculture. Then fourth one, some of the bacteria, you might have heard about the bacteria called species of Streptomycetes. Streptomycetes. You listen to you can spelling because in the notebook the spelling is different. Given the spelling is there in the Streptomycetes. Streptomycetes bacteria produce important antibiotic called streptomycin. You might have an antibiotic. There is a term we are using, medicine antibiotic. There is a meaning for antibiotic that we can discuss later on. Antibiotic. Bacteria will produce, you might have streptomycin, terramycin, penicillin. Penicillin is not by bacteria. Penicillin is produced by fungus that we can discuss later on. So there are certain life-saving medicines are there. We call it as antibiotics. So the Streptomycetes bacteria will produce the antibiotic called streptomycin. So bacteria are important. Then more importance is that some of the bacteria are used in the curing of tobacco. See, curing of tobacco industry, leather industry. They are used for various leather industry. In various industries are there. The curing of the leather for making the shoe or leather bag. Leather industries are there. Animal skin, leather is converted into bag or shoe like that. Various ornaments, materials are made by leather of animals. So there is an industry, leather industry or tobacco industry. Tobacco products are produced. Tobacco, curing of the tobacco. You know that tobacco in Kasaragod district, tobacco is cultivated. So after cutting of the harvesting of the tobacco, it is kept as bundles, one above the other for 1 month or 2 months. So that is called they are kept in bundle in anaerobic condition, covering by paper or like that plastic. So they are curing, the processing. So after 1 month, the bacteria will make it is the actual correct taste of the tobacco get by the process. Curing, the processing of tobacco, processing of leather. So like that, leather industry, tobacco industry, some of the bacteria are useful. So bacteria are useful to human beings. They are called as positive effect of bacteria. Important one. A question carrying 3 or 4 marks. Then what are the negative side of the bacteria? What are the dangerous effect of bacteria? What are the harmful effect of bacteria? First of all, they cause various diseases. See, various diseases are caused by bacteria. For example, Salmonella typhosa. See, Salmonella typhosa and Salmonella typhosa that cause typhoid. Then another example is the Vibrio cholerae. Vibrio cholerae, which cause cholera. So like that, Salmonella typhosa, Vibrio cholerae, they are all causing diseases in human beings. So they are very dangerous to human health. Then second point, they cause diseases in plants. In plant body, it will cause various diseases. And for example, Xanthomonas citriae. Xanthomonas citriae. A bacteria called Xanthomonas citriae. And that caused a disease called citrus canker. Citrus canker. Canker means, citrus. So the lemon, small one, lemon, citrus. In that lemon, on the lemon you can see brown dot can be seen like that. Brown dot can be seen. This dot is a disease. And sometimes, normally it will not happen, but during some other season, during the rainy season, lemon is available in the market. You can see brown dot-like structures are there. This dot-like structure is called, there is a disease called citrus canker. Canker means cancerous effect. Cancer is caused by virus, but this is a cancer caused by bacteria called Xanthomonas citriae. C-I-T-R-I-I. This bacteria, Xanthomonas citriae, cause citrus canker. This dot-like structure occurring to this citrus that caused by. So this bacteria produce various diseases in human beings. They cause various diseases on plants. Then another importance is that they destroy vegetables and fruits. They destroy vegetables and fruits. Usually vegetables and fruits are destroyed by bacteria. It will decompose. So they easily decompose or destroy fruits and vegetables. Then another importance is that they poison, food poisoning. They cause food poisoning. Various fungus and bacteria cause food poisoning, and you know that food is not prepared in good condition, so that it will affect the food. Or if the food is kept without covering for few bakery items like that, is kept without covering for few hours, it is contaminated by bacteria, spoiled by bacteria, because bacteria are present everywhere in the air, soil and water. If you are keeping the bakery items or food materials without covering, they will fall in that and they will begin to multiply. So like that, bacteria destroy food material and all the bakery items like that. They destroy the vegetables and fruits. They cause various diseases in plants and animals. And these are the negative effect of bacteria. Then, so this is about this is a question carry 3 marks or 4 marks. Discuss the positive and negative effect of bacteria on human beings, or what are the harmful and beneficial effect of bacteria. How bacteria are useful to human beings. These 5 points are there. You can discuss, refer the text or some other sources, and you can find out examples and answers for that. Some negative sides are there which are affecting our health and agriculture. Okay, positive and negative of the effect of bacteria to human beings. Then one small topic is remaining there. How the bacteria reproduce. What is the type of reproduction in bacteria? Bacteria are prokaryotic organisms, so they multiply very fastly. They reproduce very fastly. You know that very simply. If you are putting a little amount of lactobacillus bacteria in milk, within 10 hours or 5 hours they will multiply very fastly. So bacteria are prokaryotic organisms, are multiplying very fastly, reproducing very fastly. What is the method? The normal method of reproduction in bacteria is called binary fission. See, bacteria reproduce normally by binary fission. What is binary fission? Bi means two, fission means breaking. Binary fission. See, bacteria, binary fission means one bacteria divides into two bacteriae. that process is called binary fission. Process by which one parent bacteria divides into two daughter bacteria. That is called binary fission. See, this is called binary fission. See, bacteria reproduce by, multiply by vegetative method, simple method of reproduction. We can discuss later on. Simple method of reproduction. Bacteria divide by, multiply by, reproduce by which method? Binary fission. What is binary fission? Bi means two, fission means breaking. So what is binary fission? One bacteria divides into two daughter bacteria. Very simply, within every 20 minutes. See, the generation time of bacteria is 20 minutes. So in 1 hour, it will multiply by 3 times. Very fastly. 20 minutes it will divide. In never 20 minutes it will divide. So you can calculate in 20 minutes 2, then 4, then 8. So in 1 hour it will become 8. So within 24 hours it will multiply very fastly like that. So bacteria divide very fastly, very simply by the process of binary fission is the normal method of reproduction. Then very later it is represented that bacteria reproduce by a process of conjugation, and it is a type of sexual reproduction. It is a type of sexual reproduction. Very rarely, very later, after the development of genetics and other branches, it is explained that bacteria do a type of sexual reproduction also, not a correct reproduction, but a type of sexual reproduction that is called as conjugation. Two bacteria will join together. One bacteria, this bacteria is called, with the bacteria F factor, there is a sex factor. We can call it as male and female, plus and minus. Two bacteria come together and they will join by the process. They will join together. That is called as bacteria with a factor called F factor is called as male, without is called female. You don't bother about that. Explanations are not required, and we will not have to explain that. Anyhow, bacteria normally reproduce by binary fission, but very rarely, if the conditions become unfavorable, bacteria reproduce by sexual method, that is called conjugation. During that, there is no male female. In plants and animals, there is male animal, female animal. In bacteria, there is no male and female. But we can call it this with F, without F, plus and minus, they will join together. That joining process is called what? That joining process is called conjugation. Joining process, male, female, plus, minus, join together. That process is called conjugation, and you will not go to the details of conjugation. You refer the process called binary fission. Is the normal method of reproduction. So that is about the reproduction in bacteria, simple method. Then comes to a smallest prokaryotic organisms are there. They are called as PPLO, a small type of organism. They are called as in the textbook. There is called organisms called as coming under the mycoplasmas. So actually, kingdom Monera include 4 groups of organisms: Archaebacteria, Cyanobacteria, True bacteria, and mycoplasma. They are the smallest prokaryotic organisms known. See, prokaryotic organisms are smaller, bacteria are smaller, but mycoplasmas are smaller than bacteria. They are organisms without cell wall. They are smallest prokaryotic organisms and they are without cell wall. Bacteria are with the cell wall. These mycoplasmas are otherwise called PPLO. Mycoplasmas are otherwise called PPLO, pleuropneumonia-like organism. P stands for pleuro. This for pneumonia. Pneumonia, P is silent. P-N-E-U, pneumonia-like organism. Mycoplasmas are otherwise called pleuropneumonia-like organism. They are the smallest prokaryotic organism without cell wall. Smaller than bacteria without cell wall. And they are parasites. They cause various diseases like witch's brew. And this is a disease occurring to coconut In our area, in the coastal area, the coconut trees are affected by these diseases. Witch's brew. The leaves will be damaged. If you are traveling to the seashore from Kasaragod to Mangalore, you can see that on the sides of the river, and we are going to the border area. The coconut leaves are usually fully damaged. That disease is called witch's broom. And the leaves are, green parts of the leaves are destroyed. Only the broom remains. That part is remaining. The middle part of the leaf remains. So that is called witch's broom. And that is a disease caused by mycoplasma. So mycoplasmas are the smallest prokaryotic organisms coming under kingdom Monera. They are smaller than bacteria. They are without cell wall. They are parasites. They cause diseases like witch's brew. They are always called pleuropneumonia-like organisms. So we completed the first kingdom Monera. So today we discussed a few points regarding the true bacteria. I will sum up again. True bacteria or eubacteria are originated along with cyanobacteria or after cyanobacteria. Based on the nutrition, they are classified into autotrophs and heterotrophs. Autotrophs are two types: photoautotrophs and chemoautotrophs. Heterotrophs are two types: parasite and saprophyte. So that is the difference. And positive and negative effect of bacteria. So we discussed the classification of bacteria based on the nutrition. Positive negative effect of bacteria, type of reproduction in bacteria, and comes the mycoplasmas. 4 topics we discussed. So listen the video very carefully, repeat once or twice, write the notes that are given to you, and if you want you can take down the teaching note also on the left side of the margin side. Then so the kingdom Monera, first kingdom is over. I think you can follow my English. And then we can, next class we can switch on to the next kingdom, second kingdom called Protista or Protozoa. Thank you.